Welcome to today's webinar. I am Kailing, the Regional Field Application Scientist of Azure Biosystems. Thank you, Philly Korea, to organize this webinar. And today's we will be looking into Sapphire FL Biomolecular Imager in vaccine research. And we're looking at how the researcher in vaccine research field utilize Sapphire Classic or Sapphire FL in terms of facilitate the entire development of vaccine in R&D for commercial or in pure research purposes. But the main application in this research is on focusing on fluorescence plate-based assays, which I will explain in a specific section in this presentation. All right. So thank you again for joining this webinar. So let's just start the presentation now. Great. So we're Azure Biosystems. This is a United States-based company. So the system that you're looking later, Sapphire FL, also manufactured there. So we have a very simple motto or slogan. Say we have want to manufacture products that combine smart and simple workflows, but with high performance and affordability with flexibilities. So you can have the confidence in your data to carry on with your research or R&D and so on. We are also a Western blot solution provider. We have equipment and also consumables for your Western blotting techniques, covering from gel system, reagents, consumable, including the imaging systems. So let us look at the imaging systems then. In Azure, we have three main imaging solutions for your general or specific laboratory needs. We have Azure Imaging Systems, okay, Chemisolo, and Sapphire FL. These are the application that you will see that all these three systems covered in total. Okay, let's look at them one by one. First, let us start with Chemisolo. So, of course, Chemisolo, you can see from the name itself, it is a dedicated chemiluminescence imaging system. Okay, and next we have Azure Imaging Systems. Here I listed Azure 600, which is our highest spec model. It can do all your general nucleic acid, what is that called? Nucleic acid applications, protein gels, colorimetric applications, chemiluminescence imaging, and fluorescence signal imaging as well. And next we have Sapphire FL. Sapphire FL Biomolecular Imager. We are moving on from a general, like medium range of resolutions in fluorescence signal detection to high resolution and advanced fluorescence detections. Okay, so it covers of different specs here. Not only we're looking at like resolution, we're looking at different depth as well. So you can see the applications that can covered in Sapphire FL. And also Sapphire FL also covers phosphor imaging that is using radioisotope labeling. So this is how the system looks like. Okay, some of you may have seen these in our some exhibitions. Okay, so when you open the lid, okay, so this is how it looks like, and it has three slots for you to input the optical module. So this optical module, we call it an uncoupled optical module design. So we designed it in a way that the module and the filter is separated. So that means when you purchase an optical module, like standard optical modules, it comes with pairs, means optical module with the filters. But with that sense, since the filter allows you to swap and mix and match, it allows us to generate a different kind of optical module for your application, like yellow fluorescence protein, PI, M-cherry, so and so forth. Okay, so this design allows you to have unlimited combination of lasers and filters without purchasing additional same set of module or filters. All right, so this is what we have new innovation compared to the previous version. So this is Sapphire FL. We launched it just last year, right? So Sapphire is FL is a confocal imaging technology system. It uses a pinhole aperture, all right? So in this laser scanning system, so you can detect only the light comes from the focal plane that you set, all right? Then you can eliminate out of focus lights. Therefore, it gives you a very good resolutions and also not having the effect from those uh, out of focus light or signals. Okay. In that sense, since we have these pinhole aperture and also different depths of imaging, right? We can able to image it at different axes or different depths or z-axis. With that z-axis or z-scan, you can generate a stacks of images that cover different depths of your samples. So here's the example of a sunflower that we scan using the Sapphire FL, okay, where you can show them in, let's say, a GIF format to showcase the signal in different depths or in a merged image like this. Okay. Next, let us look at a general specs in Sapphire FL. It has a very large field of view, 25 times 25 cm. So it can put like up to 896 well plate. Okay. And that's the size that you have on Sapphire FL. Okay. And then you can image at high resolution up to 5 micron resolutions. Okay. And then we have Z plane adjustment for samples with multiple focus planes. For example, samples with different depths, like plate or even small animals. And in small animal imaging, the system has this criteria allows you to do that. First thing, it has 5 gaseous anesthesia ports built in, okay, for your scanning of your small animal, particularly maybe a rodent, okay, mice or naked mice or very skinny kind of rat, okay, or small early stages of a rat, okay. And for to fit that, we have 4 centimeter of clearance, allowing you to put samples with that 4 centimeter thickness. So that is the two criteria that allows us to image small animal in Sapphire FL. All right. So let's look at imaging samples with depth in Sapphire FL. So let's say we have a membrane, like your fluorescent Western blot membrane. Okay. So the focus plane will be at 0 mm because you lie it flat on the glass scan bed. Okay. And if you're using, let's say, a plate, 96 well plate or 6 well plate, and then the focus plane we need to set preset will be at 3 mm. All these are already preset, and also you can freely adjust the focus in the capture software as well. Supporting a wide range of focus plane from -1 plus up to +6 mm deep into the sample. This is a focus plane or Z plane that you can set in when you scan your samples. So with this setting, we can scan a huge variety of applications or samples, covering from membrane, plates, glass slide, gel, whole organs, small animals, leaves, and so on. Okay? And the signals, the fluorescent signals are also supported with a wide dynamic range with extended dynamic range function, okay, for in fluorescent signals. So allowing you to generate a simple or single field of view image that can look at the weak signal you're able to visualize. I mean, your analysis software are able to visualize a weak signal while not saturating the high intense signal as well on the same image. So with that, all the fluorescent signals to capture in this are supported with EDR function. So the system also compatible with a third-party gaseous anesthesia system. Here's a centralized uh, in input and exhaust port that is diverted into 5 gaseous anesthesia pod that go inside the system. So you can scan your small animal while like a anesthetize them and put them into sleep and cover the lid and scan them. We also have accessories such as sample holder, like a plate holder and a slide holder. So you can put your plates or slide inside, where what it does is you can select it from the preset protocol, where it will automatically select an area for you. So without you do it manually. So this is one of the accessories that paired with the capture software in Sapphire FL. So let's say selecting a slide holder protocol will auto-populate the scan areas at the correct locations. And then you can actually then how to say set the pixel size, quality and focus and also the channels for individual scan regions. Okay, so you can scan different dyes or different samples in all these presets area scan areas. It also supported with chemiluminescence module if you have this need, okay, to image chemiluminescence signals. Okay, so it comes in a separated entity, right? And the chemiluminescence module also support densitometry analysis, okay, with a standard plate. Right. So let us look at what these specs in Sapphire FL allow us to do. So the first thing is it has very good uncoupled optical modular design. So this uncoupled design between the lasers and filters allow us to do a huge variety of combination or mix and match. With that, we develop the first or the only one, okay, a 375 or UV range standard optical module that allow you to image DAPI signal. Normally, the DAPI signal will be the nuclear stain in ICC or IHC, okay, that is needed a UV range excitation to image it, okay. And then secondly, these uncoupled optical design allow us to image special dyes such as a MegaStokes dyes and also some other dyes that is not fall into our generic RGB or near infrared like combination, which is yellow fluorescence protein, PI, and M cherry, and so and so forth. Okay, so let's look at the first one. So these are the list. This is the list of standard optical module. Means that we have a laser and filter paired together with you when you order it. Okay, so these filters, as you can see, you can not only this, there are more. Okay, all right. So these I'm just using these as an example allows you to do a lot of mix and match combination. For example, here's an example of 488 channels, the standard like a side 2 channel that we know, okay. In yellow fluorescence protein or yellow fluorescence, if you want to image of yellow fluorescence protein signals, it actually excited under 488 channel, but the emission is slightly higher. The emission range is maxima is around 534. So with that, we have a dedicated filters for that yellow fluorescence protein for the best signal to noise ratio or the best signal and resolutions. Okay, so it's different. So we call it 488 yellow fluorescence protein. So this is a good example to showcase the uncoupled optical modular design. So the filter is looking like a USB drive where you can plug in and plug out. Okay, of course, swapping out and to have these combinations. Right. So Sapphire FL is the only laser scanning system on the market right now. Have these uh, UV excitation optical module, which is fall in the range of 375. Okay, compared to other laser scanning system on the market. So we developed these under the so what is that called the uh, uh, request from our users. Okay, that they needed it to image the DAPI. So here's an example. We have this in-cell Western 96 well plate of HeLa cells in seeded in serially diluted manners, 1 to 2 dilution down the column. Okay, so the bottom row is the negative control. So they basically just stain with the DAPI stain. Okay, so let's look at the left one. So on the left, image A, this plate is image under our 488 standard optical module, which is your Psi2 channel. But because DAPI is excited under the UV range spectrum, we cannot really see the signal under 488 channels. So with that, we develop a special, uh, I'll say special optical module, which is for 375. You can see here. Okay, so with that, we are able to image or visualize the TAPI stain reliably using this optical module. Okay, so that's the first thing that we have in Sapphire FL. And the second thing will be a special dye such as a MegaStoke dyes. Okay, so what is MegaStoke dyes? Which is a dye has a very huge stoke shift. That means the excitation maxima and the emission maxima uh, is hugely separated. Okay, as you can see, compared to the normal, like let's say 2 side 3 side 5 feet C, where the stoke shift is normally within 40 nanometer wavelengths. Okay, the mega stock dies, they have more than 80 or 100 stock shift, okay, nanometer wavelength. So why these manufacturer try to develop all these stock mega stock die is because you can look at here the overlapping of the excitation index and emission spectra. So these will allow you to have the lowest or as low as possible, okay, leak between the excitation from the excitation to the emission. So you have very good signal to noise ratio. And normally these dyes, what I see is people use it in small animal imaging because they need that clean background, okay, that stands out from the rest of the background noises. And with that, we able to show you, okay, with this mix and match design, we have these 488 lasers and emission filters that range of 624 to image a megastoke dyes on that we probe on these fluorescent Western blot. Okay, so next thing, let's look at another specs of this Sapphire FL. So as I mentioned before, Sapphire FL has a 25 times 25 cm field of view, so you can put like a 8 um 96 well plate or 6 well plate, it's up to you. Okay, and then we have wide Z plane scan or focus adjustment from -1 up to 6 mm, so you can accommodate anything come like from membrane gel plates up to small animals. And with that, we can also choose a huge range of resolutions from 1000 micron up to 5 micron resolutions. Okay, right. So let's look at the samples that we can do on the system. Okay, first thing, so here's a list of the couple optical module like lasers and filters. And yes, one thing I forgot to mention. So in Sapphire Alpha, we uses PMT and APD. A combination of PMT APD is dedicated for each excitation or no, sorry, the spectra. So let's say a PMT, the best quantum efficiency is at the lower range of the nanometer wavelength, which is like from Psi2 range, okay, that we use a PMT. And APG will be the one that we dedicated from like Psi3 and above, okay? And these two are the best, okay, detectors for fluorescent signals on the market right now. So let's look at the phosphor imaging right now for radioisotope or radioactive samples. So this is a type of staining or detection method that is having the highest sensitivity, okay, which utilize on or image races that require radioactivity. So they will use the same phosphor screen many times or for many experiment. Examples which is will be like a hydrogen 3, carbon 14, phosphorus protein, phosphorus 32, that is actually how to say, which staining like a glycoprotein or like a DNA or nucleic acid and so on. Of course, these also extend to nuclear study like iodine 125 and so on and so forth. So here's example. So we have a carbon 14 image plate, okay, that is image at different resolutions, up to 10 resolutions here, okay. Of course, we can do up to 5 micron resolutions in Sapphire FL, okay. So another carbon 14Z labeled brain sections, okay, that's exposed on the image plate for 3 days and scanned at 25 micron resolutions. And it's another example that we have, which taken from one of our user in nuclear study. They look at the iodine 125, actinium 225, and so on and so forth. Okay. So Sapphire FL is dedicated in fluorescence scanning. So the FL stands for fluorescence and flexibility. So of course, let's not forget the very basic application of fluorescence imaging, which is on fluorescence Western plot. So on here, we can do, at the same time, we can scan for three signals, three channels, but we can do a hot swap to image the four channels. So you can do a fourplex fluorescence Western blot imaging on Sapphire FL. Okay, right. Also, the fluorescence signal imaging also extended to your gel imaging, where to visualize your fluorescence fusion protein like GFP, also and so forth. Okay. And of course, you can also image a Comanche blue stained gel that is imaged under us. This is like near infrared 700 channels. Okay, so let's look at the resolutions here. So we are scanning these glass slide having the mouse lung tissue stained with two proteins, okay, vascular endothelia, and also smooth muscle actin at different channels, all right. But this is scanned at 5 micron resolutions. As you can see, because of 5FL, we're able to give you like 25 by 25 cm. We only can give you a 1 cm or in cm kind of scale bar. This is what it can cover. But it is not a microscope. It's a different thing. Even though it has confocal imaging technology, but it is not a microscope. Just want to clarify here, you we are not expecting you to see anything that is like within the cells in that kind of level of magnifications. For that purpose, you would need to go to certain like a confocal microscope and so on and so forth. But what Sapphire FL able to provide you is a single field of view scan or an image of your entire glass slide, a whole plate, a full gel, or whole blot, that kind of purpose. Okay, so it is very good for high throughput screening. Either you do a one preliminary screening or screening before you go for like confocal microscopy, or you want to have a standardized output of image for all your samples. Okay, so for this, let's say normally we have this in manuscript or presentations where you have these sections of area that you want to look at, and this I would recommend to replace it with image capture under confocal microscopy. Okay. Right. So with that purpose, we can like do scanning at 5 micro resolutions, okay, and 10 micro resolutions. Let's look at something we call the tissue array. So tissue array, this is a tissue array scan at 10 micron resolutions. Okay, we're looking at these samples. They are very small. They're basically in 1.5 mm in diameters. It's very small. Okay, so this is array to scan between tumor and normal samples. And with that, to showcase you, Sapphire FL also able to scan for microarray formatted slides. Okay, so you can scan at 5 micro resolution for the best image creation and downstream analysis. Right. So this is the image I brings up again to let you see what Sapphire FL able to do in terms of generating an image that allows you to do downstream analysis, not only at 5 micron resolutions on your glass slides or tissues or your plate or membrane. We're looking at something that is utilizing a different depth of scanning here. Okay. Oh, sorry. This should be -1 plus 6 mm. Right. So first, let us look at imaging of living zebrafish. Okay, this is done by my colleagues Nicole. So we have this in Thailand. So the user is scanning zebrafish that is labeled with these two dyes. Okay, and then we have this looking at the negative control and the positive controls. Okay, so we scan at 5 micro resolutions. You can see in reality they are super small. Okay, and the focus setting is adjusted at 2 mm after we do a Z scan to pick the best uh how to say best focused image. The next thing is kiwi, okay, on fruit, on fungus plate. You can see how the fluorescent scanning able to differentiate the signals, okay, clearly without at a very minimal crosstalk, very minimal to no crosstalk basically. And of course, leaves, okay, they scan at the three channels, okay, side 2, side 3, side 5, side 5, okay, and the crop because they want to look at the chlorophyll, which is at side 3 channels here. So this is scanned at 20 micron resolutions on leaves. Okay. This image is taken converted on grayscale, so you can see visualize the signals intensity clearly, or for your manuscript publication purposes. Okay, so next one is topical leaves. So looking at the topical leaves that scan at 488 channels for GFP signals, all right, and they're at 20 micron resolutions. Okay, right, mosquitoes, okay, fed with Alexa flow 488 sugar water, so the 488 will be visualized in their stomach area, right, compared to the exoskeleton that you see in the side 3 channels. So this is scanned at 50 micro resolution. Right. So let's move on to the one that will be more relevant to today's topic, which is a cell-based or plate-based assays. Okay. So this is I think this is on 6-well plate. Oh, sorry, it's a cell counting slide. Okay, this one called is cell-based assay. Right. So this is on looking at the PI and also a total cells counting. So how they analyze this is they count the total fluorescence signals comparing between two channels. So that's how they come up with a value like percentage of viability and so on and so forth. And cell-based assay, this is on I think a 24-well plate. Okay, 24-well plate, which is looking at transfection efficiencies, screening transfection efficiencies between three transfection kits. So here you can see on yellow fluorescence protein, we can signal, we will know that this kit has a best transfection efficiency compared to the rest of the two. Okay, so this is serve as a very good screening. I would say a very quick um confirmation step to let's say optimize a certain techniques, let's say transfections on your samples. Okay. Right, next, this is on microplate format, which is much utilized on vaccine research or the rest of the research on the world. Okay, so this is also cell-based assay, all right? So, but it's in microplate format in 96 well plate or 384 well plate. So, what people use this or why people use this format because of its high throughput characterization. Okay, so the properties of high throughput protein detection. All right, it may or may not be protein detection, but as long as it is emitting of fluorescent signals. So Sapphire FL here, it doesn't actually limit it to what kind of signal, but as long as within the fluorescent signal range and also the phosphor radioisotope, then you can image it. All right. So how people use this is what you can do with such a format is firstly, looking at the protein phosphorylation and signaling, compound screening for your active compound, or just looking at drug screening, or let's say an anti something a property screening, all right, RNA or siRNA library screening, quantification of viral load on your cell lines, okay, or looking at screening of monoclonal antibody clones, cell proliferation studies, functional characterization of mutant receptor. So this is more about like in cell Western that we talk about, or we're looking at cell receptor trafficking or internalizations. So we're looking at the expression on cell or in the cell expression, and also siRNA knockdown of cell surface protein. Okay, so this is on microplate format, but you can utilize it in different manners, looking at on cell expression or in cell expressions. Okay, or anything that is showing that fluorescent signals. Right. One example will be the in-cell Western that we produce in our laboratory. So we have plate focus setting M plus 3 mm. So this is an HeLa cells that is seeded in one to two directions that we're looking at here. This is a total cell stain, which is still the best and lower risk kind of normalization control for your cell-based assay. So we're looking at near infrared range because it gives you the best signal to noise ratio. Okay, looking at this gap DH and so on. So this is the merge image of them. Okay, right. So performing this will in Western on 96 or 384 well plate. Like I mentioned before, it will give you an accurate measurement on intracellular or on cell protein expression in situ. It's on the cells or in the cells. So this is very high throughput, right? You can do multiple simulations, multiple screening, endpoint, protein of interest, or replicates on a single plate because we have 96 or 384 wells. So can you have like for one group you have may have like five to more than three data points for sure. Okay. So it offers a very further improvement on high throughput data generation. Right. So here's example. We have a kit also called Azure Western kit that he's do we have this optimized with Sapphire FL. So this kit contains a permeabilization solution, blocking solution, and total cell stain, and the secondary antibody, which is in near infrared range, near infrared 700, 800. So you need to provide it just a sample, and of course the primary antibody. Okay, and then for that you can do your in-cell Western. Okay. And then here's a protocol on how to do. It's just the basic of fixing, incubate, wash, and incubate secondary wash. That's it. All right. Okay, moving on. Sapphire FL key feature. Okay, let me mention again. Sapphire FL is equipped with high sensitivity, okay, highly sensitive PMT and APD detectors. Okay, they're still the best detector for fluorescent signals on the market. All right, and then we have this uncoupled optical design or optical module that you can customize and you can change it yourself. Okay, you can freely change it, swap out, change the module, or swap the filters as you wish. And then we have a wide adjustable Z plane from -1 up to +6 mm to look in the focus plane into the samples. Resolution choice from 1000 up to 5 micro resolutions, and of course 5 gaseous anesthesia port for imaging live animals. Okay. Here's a gaseous anesthesia port you see again. Right, let's look at the imaging of small animals. Okay, so the imaging of living small animal not really necessary looking at rodents. Okay, we're looking at a huge variety of samples here. So anything that you can image them in the live format. Okay, so you can study a lot of things like application in terms of cell migrating, okay, preclinical drug testing, trafficking of the compound or certain thing or dyes over the time, or localization of your protein of interest or certain thing. Okay, so here we're looking at mouse as a very or rodent as a general example of living small animal imaging in Sapphire FL. Okay, so we have here, this is a monitoring of red fluorescence protein uh, containing tumor progression in mice. Okay, here's we can give you the example of tumor measurement. Here is the confocal imaging of these living rodents, and we have scan it in 100 micron resolution. You don't need to scan them at 5 micron resolution, so 100 micron resolution is more than enough. Okay, and then we put them in the G file so you can see the expressions or the shape of the tumor across a different focal plane. Okay, right, here's also how it looks like on 11, days 11, scanned at 100 micron resolutions. These are the size of tumor. Okay. Tumor progressions across different time points. So it's good for monitoring tumor growth or tumor progressions while not sacrificing too many animals at different time points. So you just have one set of animal to do a full time point imaging, and then you can do ex vivo dissected tumors to actually tally the results that you see in the in vivo scanning. Okay. Right, so here's another example is PEC5 labeled mouse tumors scanned at 200 micron resolutions at focus setting 1.55 mm. Okay, right. So let's look at the vaccine research. Okay, right. So in vaccine research here, I will use publication example to showcase how our user or Sapphire user they utilize the system for that purpose. It's mainly on plate, okay, 96 or 384 well plate, right? So the first example you see here, which is a chikungunya like replicon particle based antiviral drug assay to develop their vaccine, okay? So what they do normally they use in here will be like fluorescently labeled focus reduction, neutralization test like FRNT or plate-based microneutralization assays. But here's example, they are not really looking at microneutralization. I think it's type of them, but we're here looking at here, looking at this. This is actually an infected viral cell seated in 96 well plate. Okay, so they're looking at antiviral activity on the suramine and also 6AU test, right? They scan in this 96 well format. Okay, so what they're looking at is actually their EGFP reporter on the viral cells. Okay, so this is the first example that you see on vaccine research. All right. So they're looking at here. Okay. Right. Second, which is on determination of antigen concentration on the microplate. So you can see they are consistently using in microplate format. All right. So it's also in vaccine and antibody kind of research. Right. So this is on a coated antigel. All right. So they see that with different kind of polyclonals um, this is nanoparticles to look at the signals. Then they quantify the fluorescent signals in groups, okay, generated by the secondary antibodies, which reacted with the target proteins, right? So there's some examples. Right. Next thing will be identification of your monoclonal antibody specificity. So this is what they use is indirect immunofluorescence assay and also fluorescence Western blotting. They're looking at the monoclonal antibody specificity, okay, which they also paired with their here with their Western blot result here, top at column A. Also, second, fluorescence protein immunoblotting or fluorescence Western blot, and this is the one that mainly used in this group of user, which is a microneutralization activity, okay, where they publish in Cell Host and Microbe during I think the COVID time, all right. So what they call this is focus reduction neutralization test. Okay, I am not a vaccine person or a virology person, so I may not be the best person to explain this, but I will let you go through what they actually do. Okay, basically this is for COVID, checking, look at the COVID, the best antibody in development, and or looking at the response in that sense, right? So the second paper they published from the same group user in Nature Com. Okay, so they also it's the same set of technique which is FRNT and fluorescent Western blot to looking at the uh, monoclonal antibody activity against SARS-CoV-2. Again, live virus microneutralization assays, okay, against that SARS-CoV-2, right? And then again, live virus microneutralization assay, that yep, using colorimetric signals. And this is not on fluorescence then, this is on colorimetric signals. Yeah. So the previous one, they are using fluorescence signals in that sense, right? Okay. Again, another one, live virus microneutralization assays. Okay. So you can look at the publications, how they prepare this. They have already have a standard protocol to do that. All right. And then I think D is the example of how they look at it. Okay. Right. Next thing on Sapphire, what you can do is ex vivo tumor imaging, which are looking at trying to develop a vaccine that against a tumor, okay, which is tumor vaccines or cancer vaccine development. So this is also one of the example application that Sapphire FL able to do. Also another thing is RNA gel shift assay, okay, and of course chemiluminescence signal detection using the chemical luminescence module. Okay, right. So one thing for you might actually wondering why the users in vaccine research, mainly the application other than the tumors and the frozen specimen blot, why they like to use this in a plate-based format. It's all because in Sapphire FL, you only need to take 7 minutes to scan a 96 well plate, okay, or at 100 micron resolutions. So at 100 micron resolutions, 196 well plate only takes you 7 minutes to scan. So the time taken to scan and then in the capture software allows you to, let me pull up this one, right? So let's say if you're using the plate holder or you want to put just occupy the whole glass bag scanner. So you can put as much as you want to fill all them in your 96 well plate, and you just need to select or dedicate or uh, key in, okay, what kind of channels or scan speed or even the focus and pixel size, more importantly, pixel size or resolution here for each for these individual plates. Then you just click like let's say auto scan, and then you can just let's say go for your lunch or do it overnight and go home and have a rest. And this 7 minutes is very quick for 100 micron resolutions. Of course, if you're using something like a 384 well plate, then you will need to take up to maybe a 50 or 25 or 20 micron resolutions or 5 micron resolution to be able to see the wells nicely. So this is one of the best thing that in Sapphire Alpha is quick enough. It's quick enough and reliable enough for you to generate high throughput data. So with that purpose, why in vaccine research, our user in vaccine research field, they mostly use it in a plate format, a fluorescence plate-based assays. Either be something like an in-cell Western or something like this, which is looking at the reporters on the tested cell lines, or something like something like this, which is what is that called? Focus reduction micronucleation neutralization test. So these are the main application in vaccine research that utilize in our they utilize from in our Sapphire FL users. Okay, right. Okay, this have gone through. All right, just a recap. Sapphire FL is a very good, is an ultimate biomolecular imager with flexibility. Okay, so it is a very advanced and high resolution scanning for fluorescent signals and also your phosphor imaging detection for your radioisotope labeling and also like nuclear study as well. Okay, so it comes with these uncoupled designs, so you can mix and match. Okay, your lasers and filters and fret not, we also have a pad, which what we call standard optical module. They have come in pad, so you can just image, let's say, a DAPI using a 375 standard optical module or yellow fluorescence protein using this standard optical module. But let's say if you, let's say in like chemistry department or certain kind of you want to develop a certain kind of dyes that do not comply to a fixed, let's say excitation and emission wavelengths that you can see on the market, then this is what Sapphire FL able to provide you because we can swap the filters. Okay, with that you can do a combination with a unique lasers and filters or excitation and emission wavelength that you don't see on the market, that you cannot really find the imaging system able to visualize it on the market. So this is what Sapphire FL able to provide above the rest of the laser scanning system. Okay, and also equips with resolution image up to 5 micron, and then Z-plane focus adjustment to scan in depth into your samples like a zebrafish or small animals, okay, or plate, all right. And then of course in small animal there will be a 4 cm clearance and 5 gaseous anesthesia port, mostly for rodent, okay, live for rodent imaging, and then supports a huge variety of in vitro to in vivo studies. All right. So that is for Sapphire FL. And most importantly, in vaccine research, we'll be utilizing fluorescence plate-based assays. Okay. Either you're looking at fluorescent signals from the cell lines or something called FRNT, I forgot the focus microneedlization assay. Okay, right. Sapphire FL has been well received all over the world. Here's some of the representative users we installed in for these past, I think, 10 months, I guess. Right. So they are freshly newly installed. Okay, we have one in Korea right now, if I'm not mistaken. Yeah, I installed one. Okay, and then for Sapphire FL. Of course, we have our Sapphire Classic, which is a discontinued version that we launched in 2017. Okay, we have quite a few of them basically. If you look at our publication track record on our website. So if you're interested in looking at how Sapphire or Sapphire FL able to provide you what kind of application you can do, we have publication booklet. Okay, publication booklet will give you something like this. We are showing you what is the where the paper come from, what's the main results they're able to produce from using Sapphire or Sapphire FL. And then with that, we also have tons of not tons tons of application notes that they has individual dedicated or specialty assays. For example, here like by FC assays, okay, for in vitro protein ligand interactions and screenings from autophagies and the thing like an in-cell western or anything plate-based assay in this, or microarray and so forth. Okay, so please feel free to look them up. Okay, if you're interested, you can talk to our representative in Free Korea. Okay, Ban, our product manager, so he can actually provide you these informations. Right. So here's some examples of how it looks like. Is the application notes. Okay, in vivo imaging is all this not all, but a part of the data that you see that I presented before, they are all already formulated in application notes. So if you want to know more details, please feel free to look them up. Okay. So we Azure Biosystems. Okay. So we have imaging solution for your basic laboratory needs and routine and applications. Sapphire FL is our highest end system. That is our this is something we are very confident with, which provide you advanced and high resolution fluorescent scanning, including phosphor imaging scanning as well. So thank you so much for your attention. I am Kyling. Um, if you have any questions, please talk to us on. There's a chat box here. Let us know what you want to know about Sapphire FL. Thank you.